Niacin's role in insulin sensitivity and glucose metabolism is one of the more complex and paradoxical aspects of its biology, simultaneously revealing its therapeutic potential and exposing the limitations of reductionist thinking in nutrition and medicine. While niacin has been shown to influence insulin signaling, enhance glucose transporter activity, and support pancreatic beta cell function under certain conditions, it has also been linked, especially at high pharmacologic doses and in specific forms, to mild insulin resistance. This apparent contradiction has created confusion in both clinical and research communities, often leading to a blanket wariness about niacin in diabetes care, despite deeper evidence showing that the story is far more nuanced. At a cellular level, niacin, vitamin B3, modulates key pathways involved in glucose metabolism. Through its conversion into NAD+ and NADP+, niacin supports hundreds of enzymatic reactions critical to mitochondrial function, oxidative phosphorylation, and the citric acid cycle, all essential for proper glucose utilization. More specifically, NAD+ fuels enzymes like sirtuins and PARPs, which regulate insulin sensitivity, mitochondrial biogenesis, and inflammatory response, all of which have downstream effects on how cells respond to insulin and manage glucose uptake. Studies have shown that niacin can upregulate GLUT4, the insulin-sensitive glucose transporter found in adipose tissue and muscle. This is the transporter responsible for bringing glucose from the bloodstream into the cell, where it can be used or stored. Upregulating GLUT4 improves glucose clearance and insulin sensitivity, particularly in metabolically compromised individuals. In animal models and certain human studies, niacin supplementation has been associated with better glucose tolerance, reduced fasting insulin levels, and enhanced metabolic flexibility, especially in those with early-stage insulin resistance. But here's where the narrative becomes complicated. High-dose niacin therapy, particularly in the nicotinic acid form used to manage cholesterol, has been associated with modest increases in fasting blood glucose and insulin levels in some individuals. This effect is believed to be partly due to a rebound in free fatty acids, which niacin initially suppresses by inhibiting lipolysis in adipose tissue. When this suppression is lifted, as niacin's effects wear off, there's a surge in free fatty acids, which can temporarily induce insulin resistance. This phenomenon is often misinterpreted as a uniformly harmful effect, leading to the assumption that niacin is detrimental in diabetes. In reality, the picture is more context-dependent. These blood sugar elevations are typically modest, transient, and often clinically insignificant when balanced against niacin's broader metabolic benefits, such as improvements in lipid profiles, reductions in inflammatory markers, and potential NAD+ replenishment. Moreover, in longer-term studies, the glycemic effects tend to stabilize as the body adapts, especially when niacin is introduced gradually or used in non-flushing, low-dose regimens. The form of niacin also matters greatly. Niacinamide, the non-flushing form of vitamin B3, is less likely to cause shifts in lipid metabolism and appears to have a neutral or even beneficial effect on glucose control. In some cases, it has been shown to protect pancreatic beta cells, reduce oxidative stress in diabetic tissues, and prevent the progression of type 1 diabetes when used early, particularly in autoimmune-prone individuals. This again suggests that niacin's metabolic effects are not uniformly positive or negative, but highly dose-, form-, and context-dependent. Despite this, niacin has been largely written off in diabetes care due to its association with glucose elevation in high-dose lipid therapy. But this dismissal ignores the larger biochemical role of NAD+ in metabolic health. NAD+ levels are consistently lower in individuals with diabetes, obesity, and metabolic syndrome. This deficiency contributes to mitochondrial dysfunction, impaired insulin signaling, inflammation, and oxidative stress. Niacin, as an NAD+ precursor, addresses this upstream problem, something that glucose-lowering drugs do not directly do. In truth, the dual role of niacin in both promoting and impairing insulin sensitivity is not a flaw, it's a mirror of metabolic complexity. It reflects the reality that nutrients are not drugs, and that their effects are modulated by cellular context, dosage, form, and existing metabolic state of the individual. When used appropriately, niacin can enhance metabolic flexibility, support mitochondrial resilience, and provide systemic benefits that far outweigh its minor and manageable impact on blood glucose in specific cases. It's time to evolve the conversation around niacin in diabetes and metabolic health. Instead of simplistically labeling it as bad for blood sugar, We should recognize it as a regulatory molecule, one that can be powerful in restoring balance to disordered energy metabolism when used intelligently. It deserves a place in nuanced, integrative metabolic care strategies, not just for its NAD+ connection, but for its capacity to modulate inflammation, oxidative stress, and cellular energy, all of which are at the heart of insulin resistance and glucose dysregulation. In short, niacin is neither hero nor villain in diabetes. It is a misunderstood tool. And like any tool, its power depends entirely on how it's used.